Hi, everyone. Today, we're going to take a look at a recent preprint from Sheverman colleagues that presents a new high-resolution atlas of menstruation. Using a novel mouse model and spatial genomics, their work really challenges a long-held view of the endometrium. It reveals a highly organized wave-like process of cellular change where we previously assumed a much simpler, layered structure existed. So let's examine how they accomplished this. So here's our roadmap for this discussion. We'll start with the innovative mouse model that made this research possible in the first place. From there, we'll review the spatial transcriptomics methods they used, before diving into their findings on fibroblast and decidual cell organization. Then, we'll examine the evidence for a cellular maturation gradient, and wrap up by discussing how these discoveries are revising our fundamental understanding of endometrial architecture. To study a process as dynamic and hormonally complex as menstruation, you need a controllable and replicable in vivo system. For a long time, the absence of such a model has been a pretty significant barrier to progress in the field. And this is the solution the researchers came up with. The X-men's model is a transgenic mouse that utilizes chemogenetics. What this means is that specific proteins have been engineered into the mouse uterine cells that can be activated by a designer drug, one that's otherwise inert. This lets the researchers precisely trigger and synchronize decidualization and the menstrual-like shedding that follows, and it does so in a way that faithfully recapitulates the key anatomical and transcriptional hallmarks of the human process. This gives them unprecedented temporal control to study the process moment by moment. Okay, so having this model is a huge step forward. But the challenge still remains to resolve what's happening at single-cell resolution while also preserving the tissue's original geography. And this is where their sophisticated methodological approach becomes really critical. This slide here outlines their workflow. It all starts with collecting uterine tissue from the X-men's model at very specific time points after induction. Next, they use a technique called slide tags, which assigns a unique spatial barcode to each cell nucleus right on the tissue slide. After that, single-nucleus RNA sequencing is performed to quantify the transcriptome of every single one of those cells. And the final step is all computational, integrating the spatial barcode data with the transcriptomic data to reconstruct the tissue in silico, linking every cell's gene expression profile back to its precise physical coordinates. So let's define slide tags a bit more precisely. You can think of it as assigning a unique spatial coordinate, almost like a zip code, to each nucleus before it's removed from the tissue for sequencing. This technological advance is really what allows the researchers to create these high-resolution maps, directly correlating a cell's identity and its functional state with its location and neighborhood within the tissue. Now, this whole process generates an incredibly high-dimensional dataset. To analyze it, they used SCVI, which is a tool based on a variational autoencoder. Its function is really twofold. First, it reduces the dimensionality of the data, letting them find the most important patterns in gene expression. And second, and this is crucial, it corrects for batch effects. These are the non-biological, technical variations you get from processing different samples at different times. This computational step is essential for making sure that the differences you see reflect true biology, not just experimental noise. The final key computational concept here is pseudo-time analysis. This technique lets you move beyond just static cell type clustering. By ordering cells based on the gradual progression of their gene expression profiles, it computationally reconstructs a continuous biological process from these discrete snapshots. This allows the inference of a developmental trajectory like differentiation, and it creates a quantitative timeline that can position individual cells as early or late stage along that specific path. All right, with those methods established, let's proceed to the first set of results. The team started by mapping the tissue's architecture in its baseline, uninduced state, with a particular focus on the stromal fibroblasts. And right away, the spatial analysis revealed that the endometrial fibroblast subtypes, or EFibs, are not just some homogeneous, randomly distributed population. Instead, they form distinct, spatially organized layers within the stroma, which indicates a pre-existing structural organization. This slide details that layered organization. The E5-1 cells are found in the basal region, right up against the myometrium. The E5-3 cells are on the opposite side, directly beneath the luminal epithelium. And located between these two layers are the E5-2 cells. They also found a fourth population of proliferating fibroblasts, E5P, that was co-localized with that E5-3 layer. So this demonstrates a very structured cellular geography even before decidualization is induced. So this organized baseline provides the context for the mean event.
We're now going to examine what happens when decidualization, that's the transformation of the endometrial stroma, is induced in the X-men's model.
The transformation, it turns out, is not a diffuse or chaotic process. Instead, two new decidual cell populations, which they call DEC1 and DEC2, emerge. And critically, their appearance isn't random. They form a highly organized and very distinct pattern of concentric rings within the stromal tissue. As the process unfolds over time, this pattern evolves. A third decidual cell type, DEC3, appears at the very center of those pre-existing rings. The entire decidualized structure then continues to expand radially outwards, all while maintaining this concentric organization, almost like a wave propagating from a central point. Now, the terminal phase of this process is tissue breakdown and shedding. The analysis identified an additional decidual cluster, DACP, which was mainly observed in the menstrual phase samples. The transcriptome of these cells is enriched for genes related to cellular stress and apoptosis, including the pro-apoptotic factor BAX. This suggests that the DACP cluster represents cells that are undergoing programmed cell death as the endometrial lining is shed. This highly ordered spatial and temporal progression, from layered fibroblasts to these concentric decidual rings and a central core, well, it strongly suggested a deterministic underlying mechanism was at play. And that led the researchers to develop and test a model to explain these observations. So the central question became, what is the biological process that drives this highly structured, concentric spatial organization of distinct decidual cell states? This led them to hypothesize a maturation gradient model. The core idea here is that the concentric rings don't just represent distinct cell types, but rather different stages of a single, continuous differentiation process. In this model, the outermost DEC1 cells are the least mature or youngest cells. The intermediate DEC2 ring represents a proliferative intermediate stage, and the central DEC3 cluster is made up of the most mature cells, the ones that initiated the differentiation process the earliest. And this is where the pseudotime analysis provided critical quantitative support for their model. As quoted from the preprint, when they mapped their calculated pseudotime values onto the spatial coordinates of the tissue, they saw a strong positive correlation between pseudotime and radial position. The pseudotime values, which represent inferred cellular maturity, increased progressively from the periphery toward the center. This provides strong evidence that the concentric rings do, in fact, represent a spatiotemporal wave of cellular differentiation. So let's now consider the broader implications of these findings. This new data fundamentally challenges a century-old and, frankly, simplified model of endometrial architecture. The crucial point is this: the endometrium has long been described by a simple bilayered model, made up of a regenerative basalis layer and a shedding functionalis layer. This work demonstrates that the biological reality is far more complex and dynamically structured than that. This slide here contrasts the two models quite well. We're moving from a static, bilayered view to a dynamic, multi-layered model that's defined by function and developmental time. It's not merely two layers, but a process of radial differentiation. This concentric maturation gradient creates a very precise cleavage plane at the boundary between the fully decidualized and the non-decidualized tissue, which effectively predetermines the site of shedding. In summary, this study provides a new spatiotemporal framework for understanding how the endometrium remodels itself with such precision during each cycle. It really serves as a foundational, high-resolution atlas for a biological process that is central to reproductive health. And this leaves us with a critical, forward-looking question. With this much more detailed and dynamic map of normal endometrial remodeling, how can we now better understand the cellular and molecular dysregulation that occurs in pathologies like endometriosis, adenomyosis, or certain forms of infertility? This work provides a powerful new lens through which to investigate these very questions.